Good morning, all of you. This is Anita Mani, Associate Professor at Government College for Women, Lakhan Majra, Rohtak. Today, we are going to discuss the TCA cycle, that is tricarboxylic acid cycle. We also call it citric acid cycle or Krebs cycle. Citric acid cycle is actually a series of chemical reactions used by all aerobic organisms to release stored energy through the oxidation of acetyl coenzyme A derived from carbohydrates, fatty acids, and proteins. It is second stage of cellular respiration. This process is carried out in matrix of intracellular structure called mitochondria except in bacteria. Krebs cycle or citric acid cycle is one of the most important reaction sequences in biochemistry. Citric acid cycle is closed loop. The last step of pathway regenerates the molecule used in first step. Krebs cycle starts with the product of glycolysis, which are two 3-carbon molecules called pyruvate. This molecule is acidic, which is why the Krebs cycle is also called TCA, that is tricarboxylic acid cycle. Throughout a number of reactions, these molecules are further broken down into carbon dioxide, water, and energy. Energy from the molecule is moved to other molecules called electron carriers. These molecules carry the stored energy to the electron transport chain, which in turn creates ATP. TCA cycle consists of 8 steps catalyzed by 8 different enzymes. The cycle is initiated when acetyl coenzyme A reacts with oxaloacetate to form citrate and release coenzyme A SH. Now let's study the reactions of TCA cycle. In step 1, there is oxidative decarboxylation of pyruvate to acetyl coenzyme A by PDH complex. This step is connecting link between glycolysis and TCA cycle. Oxaloacetate combines with acetyl coenzyme A to form citrate catalyzed by enzyme citrate synthase. In step 2, citrate is isomerized to isocitrate by the enzyme aconitase. This is achieved in a two-stage reaction of dehydration followed by hydration through the formation of an intermediate cis aconitate. In step 3, isocitrate loses a molecule of CO2 and then undergoes oxidation to form alpha ketoglutarate by enzyme isocitrate dehydrogenase. Along with CO2, a molecule of NADH is also formed. In step 4, alpha ketoglutarate loses a molecule of CO2 and is oxidized to form succinyl coenzyme A by alpha ketoglutarate dehydrogenase complex. At this stage, a second NADH molecule is also produced. In step 5, succinyl coenzyme A is enzymatically converted to succinate. It is accompanied by conversion of GDP to GTP and GTP to ATP by enzyme nucleoside diphosphate kinase. In step 6, succinate is oxidized to fumarate by enzyme succinate dehydrogenase. This results into formation of FADH2. As you can see that in step 7, it involves conversion of fumarate to malate with the addition of water in presence of enzyme fumarase. In the last step, malate is oxidized to oxaloacetate by malate dehydrogenase and closes the cycle. A third NADH is synthesized at this stage. Oxaloacetate regenerated here combined with another molecule of acetyl coenzyme A and continue the cycle. This is complete TCA cycle. All the steps are being represented very clearly. It is a very simple cycle to learn. Thank you so much.